guys! So today's lesson we're going to be looking at the monitoring of concentrations of ions in samples. So usually when we measure concentrations of ions, we need a way to visualize it and get all the information in one go. And the easiest way to do this is in a graph. So in this graph we're looking at zinc ions in effluent, and on the x-axis we have effluent in mils, and on the y-axis we have concentration of metal ions. So in this case it's zinc. So once we get all our data, we plot it on the graph. So say for this one we plotted the first one at a 0 concentration of zinc. We had just under 100 mils of effluent, and so on up to here. And using a graph, this allows us to visualize it and see if there's any trends. So here we have a trend that if you increase the amount of effluent from 100 to 0 to 300 and then plot the concentration of zinc ions, we get increasing amounts with increasing effluent. So as we increase the effluent, we get increasing zinc ions. So when we look at samples, we're really looking at how much is in there. And this is really important for analytical chemists because they need to allow us to have a standard of water or a standard of their products, so we know that what we're getting is what we want. So chemists routinely analyze water samples in our reservoirs to make sure that the amount of metal ions in there is within the safe range. So in particular, lead ions are important because they need to be monitored carefully. This is because that lead is a poison. It leads to retard intellectual development of young children, and also it causes brain and neurological disorders. Lead compounds used to be in petrol, and if they weren't banned, they would be released into the atmosphere by vehicle exhausts. But the only thing is that dust containing lead still is released into the environment because in old buildings and in the demolition of old buildings and renovation of them, there used to be lead paint. So as you demolish it, it breaks up the lead paint and it gets into the atmosphere. So as we talked before about AAS, it's a way to monitor the amount of metal ions in a sample. And we can look at soil samples, water samples, any sample really. It's more sensitive than previous techniques, so that's why now it's used so extensively. So the pollution of the environment and the manufacture of goods containing lead compounds is now banned. It's prohibited by law because of its neurotoxicity. The sensitive and quantitative measurements and monitoring of lead ion concentrations is essential to prevent any neurological disorders in people. Next, we're going to look at phosphate ions. So over there is a phosphate group. And phosphate is found naturally in the waterways in low concentrations. And at these low concentrations, there is no detrimental effects to the environment. It is essential for aquatic growth, but if the concentration gets too high, we can get an algal bloom or rapid algal growth, shown in the picture over there. And this can cause a lot of problems environmentally. So the surface of rivers and other waterways can be covered by algae, which can choke the waterway systems. Algal blooms are set off when we have too much nutrients. So nutrients such as phosphate groups can cause algal blooms. Environmental conditions which are favourable also can cause algal blooms. So we need warm temperatures, we need sunlight, and we need still water, because movement of water will move the algae, and so that they can't grow. So algal blooms can make the water unusable for humans and for livestock, so that means we can't drink it. It can potentially lead to permanent degradation of the river or lake, and it also can consume oxygen from the water. So if we're going to starve fish of oxygen, they can't breathe, and therefore they're going to die. It also leads to a build-up of sediments in the dams and rivers, which can reduce the flow of the water. And uncontrolled growth of algae leads to depletion of the phosphate, which is required for aquatic life. So as the algae dies at the end of the cycle, it's decomposed by aerobic bacteria. So aerobic means that it uses oxygen to carry out its functions. And that means if the algae used oxygen initially, and then now when it's decomposing, the bacteria is using the oxygen, it's going to be using all the oxygen that's usually for the fish. So we're going to get very dead waterways. So eutrophication is the process by which a water body becomes enriched by nutrients. So this can include phosphates and nitrates. And this, if it's too much in there, it can lead to algal blooms. Phosphate ions are derived from a variety of sources, and these can include sewage, farm fertilizers, and some laundry detergents. So we need to monitor the presence of phosphate in the waterways to predict if eutrophication is going to occur, and if it is, how we can stop it. So to monitor phosphate ions, we use a colorimetric method. So that means it uses light and color to analyze the sample. It's very sensitive because only 0.05 parts per million of phosphate ions in the water are required for algal blooms to occur. And to do this, we use a molybdenum blue test. So the colorimetric test here is using blue light. So a spectrophotometer, in the picture over there, measures the absorbance of light at a particular frequency. In this case, because it's a blue test, we're looking at the frequency of blue in this technique. So the absorbance of the test sample is compared to a set of standard solutions where we know the phosphate concentration in them, and then we can plot a calibration curve like we did with the AES technique. So once we've plotted a calibration curve, we can analyze our sample, figure out how much we have absorbed light there is, and then look at how much concentration of phosphate ions is in there. So next we're going to look at ions. So these comprise a very small amount of the body mass, only 0.008%. It's known to be a trace element, and it's important for a variety of proteins in the body. So proteins such as those needed for DNA synthesis and cell division need iron. Iron is also complexed with hemoglobin, which is in red blood cells, and it's necessary for the oxygen transport in the blood because it binds oxygen and transports it to areas where you need it, such as your muscles. Iron is used to help produce connective tissue in the body, also some neurotransmitters to let your brain function, and also to maintain a healthy immune system to prevent you from getting sick. So an adequate supply of iron, we need to make sure that we get it from our food because our bodies continually get rid of iron through a variety of processes. And this includes urination, defecation, and sweating, as well as the sloughing off of cells, which is the getting rid of skin cells. This is not a usual occurrence, but bleeding also contributes to more loss of iron. So because we're losing a lot of iron from all these processes, we need to make sure we compensate. So usually what we do is we eat our sources of iron. So in our food, we should approximately consume 18 milligrams of iron a day. Otherwise, we'll be deficient in iron. So scientists need to be able to monitor the levels of iron in living things to determine if we have a deficiency or not. If we have a deficiency, then our normal processes can't occur and we can get really sick. We also need to be able to monitor and measure the levels of iron in the food so we know how much of that type of food we need to eat. Next, we're going to look at fluoride. So fluoride is found in our water, and these ions are analysed for health reasons. So we need small amounts of fluoride in our water so we can help harden our teeth. Places that don't have fluoride in their water, their drinking water, the people tend to have softer teeth and they get more cavities. But the thing is that we need to make sure that the levels of fluoride in the water are kept in a tight range. So between 0.9 and 1.5 parts per million. If it's over 1.5 parts per million, then it can have damaging effects to our teeth. And this can include the mottling of the teeth. So now for some questions. So question 11, we're going to need to outline the need to monitor the ion you have studied. So the verb here is outline. So I'm just going to circle that. And that we need to monitor. Why do we need to monitor an ion? This one doesn't specify an ion. So what we've done is we looked at lead. So lead is a toxic heavy metal which can bioaccumulate and biomagnify in the food chain. So what this means is that bioaccumulation is the process by which lead, in this case, accumulates in the body. So either by eating other animals that have lead in them. And the biomagnification is the process by which, as you go up the food chain, because the smaller animals have small amounts of lead, we then eat them, accumulate the lead in them, and then go up higher in the food chain. So the top ones that eat the most amount of animals have the highest amount of lead in them. Lead is also released into the environment from mining, lead-based paints, pottery, stained glass, plumbing materials, and battery manufacture. So pretty much we need to make sure we monitor lead ions to make sure we don't get toxicity in our system. So question 12, what does AAS stand for? And briefly describe how it works. So here we have the question, what? We need to describe what AAS stands for. And the second half of the question is make sure you describe how it works. So the first part of the question, what does AAS stand for? It stands for atomic absorption spectroscopy. Next, we need to describe how it works. So it involves measuring the amount of light absorbed in a sample in order to determine the concentration of ions in the substance. So AAS is a sensitive technique that relies on absorption of light to determine a concentration. Next, we have question 13. So make sure we identify the name of the graph obtained by plotting the absorbance against or standards against the concentrations. So here we have a graph usually that we produce at the beginning of analyzing a sample with AAS. So what we usually do is we get a known sample of a known concentration of our sample. So here we'll look at, say, we have concentration on our x-axis, and on our y-axis we plot absorbance. So if we know the concentration and then we measure the absorbance, we can then plot it. And then we do this for a range of concentrations that we know, and then measure the absorbance. So this is a standard. We then can draw a line through it to get the trend, and then from this we can then check our unknown sample and look at the concentration and the absorbance to determine it. So this graph is then called a calibration curve. So a calibration curve can be used in AAS as well as other techniques when you don't know the concentration in your sample. So question 14, name 3 trace elements in the body whose presence can be monitored by AAS. So in this question we only need to name 3 trace elements. So 3 trace elements which can be analysed by AAS include zinc, it includes iron and magnesium. Next, we'll look at question 15, which is list the processes by which our body loses iron. So here we only need to list them, so we don't need to explain or anything, and how our body loses iron pretty much. So to lose iron from our body, we urinate, defecate, sweat, and get rid of skin cells, or sloughing off of skin cells. So today in our lesson we covered different ions which need monitoring in our environment. So this included phosphate ions to make sure that the waterways don't get choked with algae. We looked at lead ions because it causes neurotoxicity in humans and causes learning defects. We looked at iron because it's important in a lot of biological processes. So using all these, we can answer a lot of questions about trace elements that are required inside our environment, as well as the detrimental effects if they're not monitored.